right. Well, good morning and welcome to today's session of Strategic Farming. Let's Talk Crops. We're glad you could join us for today's session on how we adapt to insects overcoming previously reliable tools. Basically, insect resistance is what we'll be talking about today. These sessions are brought to you by University of Minnesota Extension and with generous support from the Minnesota Soybean Research and Promotion Council, as well as the Minnesota Corn Research and Promotion Council. I'm Anthony Hansen, an extension educator in integrated pest management, and today we kind of have a split session between this soybean and corn world for insect issues. First up, we'll have Dr. Bob Koch. He's our soybean extension entomologist with the University of Minnesota. And then later on, we'll have Dr. Fei Yang, our corn extension entomologist, also with the University of Minnesota. So with that, I'd like to turn over to our first speaker, Bob Koch, if you want to start off with you and let us know just what's going on with soybean insects and resistance, whether it's insecticides or other issues coming up.
Okay, great. Yeah, so I want to provide some updates on the soybean aphid, in particular with the insecticide resistance, and then some updates on soybean gall midge, which is, again, a new pest that we're dealing with in Minnesota. But no resistance issues there, just more of a general update. So soybean aphid, I think we're all pretty well aware of this pest, right? You see a female aphid giving live birth, and their short generation times, high reproductive potential can lead to very heavy levels of infestation. We're not exactly sure why, but infestations have decreased in a lot of the states to the east of us. So our colleagues in Ohio, Michigan, Indiana, Illinois rarely deal with soybean aphid anymore. However, in Minnesota, parts of the Dakotas, northern Iowa, we still have pretty consistent infestations of soybean aphid, and it remains our most important insect pest in soybean. And our management of this pest still relies mainly on insecticides. Right, so we still got some organophosphate use, pyrethroids, neonicotinoids, and then some of the newer subgroups of sulfoxamines, butanolides, and pyropenes. And we'll talk a little bit more about those a little bit later on here. But for the pyrethroid insecticides, I just want to give kind of a quick recap on the kind of historical overview of the situation, where we did some studies in southeastern Minnesota, just standard insecticide efficacy trials. Bruce Potter was doing similar work in southwestern Minnesota and Lamberton. We pooled all that data together to look at how the pyrethroids, in this case it was lambda-cyhalothrin, how it was performing over time at these two locations, Rosemont and Lamberton. And then the y-axis, or the vertical axis here, is percent control. So you see from the beginning, 2025 to 2014, we had nearly 100% control. So the pyrethroid insecticides were doing what they should be doing. However, from 2014 onward, that percent control decreased very rapidly. And that change coincides with when we started getting reports from growers of the pyrethroids not working as well as they used to, and when we started doing more detailed laboratory work to further confirm resistance. So we've been seeing resistance to the pyrethroids across multiple locations in Minnesota, into adjacent states and provinces, and across multiple years now. Want to show you some of our data from last summer, where we're still doing these kind of standardized small plot insecticide efficacy trials. This was again at Rosemont. We used later planted soybean, and we sprayed at the R3 growth stage when we had about 60 aphids per plant. So the data I'm going to show you will be on the graph like this, where we've got on the vertical axis the mean number of aphids per plant. Across the bottom, we've got different insecticides, with first an untreated, so no insecticide, then Warrior, which is a pyrethroid, Indigo, which is a pyrethroid and neonicotinoid mix, and then Safina, which is one of the newer insecticides, and then Renestra is a mixture of Safina with a pyrethroid, and then we've got Transform, which is one of the newer insecticides, and then a mixture of that with a pyrethroid, which is Ridgeback, and then Savanto, So again, we sprayed this experiment when there were about 60 aphids per plant. And then we came back after 1 week, and we see that in the untreated plots, aphid numbers increased to over 100 aphids per plant. And the pyrethroid or warrior treated plots increased similarly. And I guess going back, then you see for all those, the indigo and all those other newer insecticides, very, very low levels of aphids. So we got good control right off the bat with those. And then by 2 weeks out, we see numbers in the untreated plot beginning to decrease naturally, whereas numbers of aphids in the warrior or pyrethroid treated plots are increasing. And then by 3 weeks, that trend continues. And across all those newer insecticides plus that mixture of the pyrethroid and the neonicotinoid, we maintain good control. So just to kind of recap, I would say in this experiment, an insecticide application wasn't needed, right? Because those populations naturally decreased. Plus, we sprayed when it was only 60 aphids per plant, which is well below the economic threshold of 250 aphids per plant. So that's that trigger point where we would recommend an insecticide application being made. We sprayed this plot at this time because we needed to get some data. Summer was running short. So we set up the trial, but I think it's a pretty good case study that all infestations don't continue to necessarily increase. What I think also is important to see is pyrethroid resistance was still clearly out there in this population, and that applications of the insecticides can sometimes make situations worse. So by the time we got out to 3 weeks after infestation, there were 10 times more aphids in those warrior treated plots than in the untreated plots. We're going to talk a little bit more about some of these mixtures in a few slides, but I just want to point out that I've been seeing some inconsistent control with some of these pyrethroid neonicotinoid mixtures for soybean aphid. They worked generally pretty well in this experiment statistically. At 2 weeks out, the control wasn't as good as some of those other newer insecticides. And then again, with those newer insecticides, we see that they all perform very well against this pyrethroid resistant population. One thing I want to comment on though is these mixtures like Renestra and Ridgeback. If you've got Safina and Transform already working well against aphids, adding the pyrethroid or some other insecticide to it probably isn't going to provide much of a benefit there. I think these mixtures could provide some value if there are infestations of different kinds of pests occurring at the same time, but in Minnesota, that's typically not the case. I mean, we're always going to have different kinds of pests in our fields, but kind of economically concerning levels of multiple pests is not always a very common situation. So I'm not a huge fan of the mixtures. But speaking of the mixtures, they are pretty widely available, and almost all of them, if not all of them, at least listed in this publication that we produced, contain pyrethroids. So remember, many of the soybean aphid populations have resistance to pyrethroids. So if we're spraying infestations with a mixture that contains a pyrethroid, we're probably not going to get much benefit from the pyrethroid component of that mixture. And if we look back to 2024 at a different experiment that we performed at Rosemont, another insecticide trial. In this case, we had an untreated check. We had leverage, which is a pyrethroid neonicotinoid mixture. And then we had Savanto, Safina, and IKI is a flunicimid. It's not labeled for aphids, but I think the company is trying to pursue a label. What I want to point out here is by 1 week or 8 days after treatment, the leverage statistically was not different from the untreated, whereas the other insecticides significantly reduced the infestations. However, by 2 weeks out, 15 days after treatment, those leverage treated plots, again, the plots treated with a pyrethroid neonicotinoid mixture, had significantly more aphids than the untreated plots. So this is kind of another case study of what can go wrong with insecticide applications. In the 2025 experiment, we saw our warrior or pyrethroid treated plots having more aphids than the untreated plots. And then the year prior to that, we saw a similar situation with the pyrethroid neonic treated plots having more aphids than the untreated.
So Bob, we got a quick question for you related to that,
Yep.
or maybe a couple. One is, based on other trials you've done with those foliar neonicotinoids, just alone, do you think it's just the poor pyrethroid control driving things, or is there something in the mix going on? Or when we talk about mixtures, sometimes they reduce the rates. Is that maybe what's going on, do you think?
Yeah, so I mean, part of the product isn't probably isn't working because of the pyrethroid resistance, and then that other part, if it's a neonicotinoid, from what I've seen, the foliar neonicotinoids typically don't provide as consistent control of the soybean aphid. I think that was some work from Bruce Potter, Ian McCrae, a number of years ago, and some other folks. And if you think about the different products available for soybean aphid management, we don't really have any pure neonicotinoid products that are getting much use for this pest, right? Um, Sometimes products, when there are mixtures, they'll have reduced rates of some of the actives. I haven't checked that for all these, but sometimes that's a situation. And um, sometimes these mixtures, at least for insecticide resistance management, aren't the best strategy. It's a trickier strategy for insecticide resistance management than it is for herbicide resistance management. And speaking of resistance management, I just want to point out that we've updated our multi-state publication on managing pyrethroid resistant soybean aphids. I think the team will share a link to this in the chat section. And kind of the key points here are that we want to be scouting our fields and using the economic threshold to determine when to spray. We're still standing behind the threshold of 250 aphids per plant as a pretty conservative trigger point for deciding when that insecticide application is needed. If we're going to spray, we want to make sure we're doing it right, having proper rates, volumes, pressures, all those things. We want to scout our fields after spraying to make sure that the insecticide did its job. I don't think we can just assume anymore that insecticides are always going to work. Here I listed 3 to 5 days. If you're using a product like Safina, be aware that those aphids don't always die right away. That product stops the aphids from feeding, and you end up with kind of zombie aphids lingering on the plants, slowly dying, but they're not feeding anymore. So from a plant protection perspective, an aphid that's not feeding and not reproducing is as good as a dead aphid, but you might see aphids lingering longer on Safina treated plants than plants treated with some of the other insecticides. And if you need to spray again because of a failure or a reinfestation, make sure you're alternating to a different insecticide group. So pay attention to those group numbers on the insecticide labels.
Speaking of that,
Uh,
Bob, another question that comes up sometimes is on the organic insecticide side of things. You might hear about pyganic. Do you want to comment on that one a little bit, how that falls into this scheme framework here?
yeah, so pyganic's going to be kind of related to the pyrethroids in that same group or subgroup. The pyrethroids are essentially a synthetic version of what pyganic is made out of. Our organic growers have pretty limited options. That's one of the few options available. I think it can be effective if you're getting good contact and coverage with the of that product to good coverage to get contact with those aphids.
Right. But that's still in a group three, isn't it? So how does
Yes.
that
Yep.
kind
So
of
in
fit
that
in
same
with
group or
that?
subgroup.
Yep. Okay.
So I'd suspect if you got a population that's resistant to pyrethroids, pyganic probably won't work very well. I suspect there'd be cross resistance there, but I don't think anyone's tested that yet. So kind of wrapping up the soybean aphid end of it, just pointing out again, soybean aphid continues to be a pretty significant pest in Minnesota, even though its infestations have petered out in some other states. Our management of this insect is challenged by insecticide resistance to the pyrethroids. We don't have all that many other chemical tools out there, so we do need to be careful and thinking about insecticide resistance management to try to preserve the chemical tools that we do have. And unfortunately, we just don't really have many other non-chemical tools available to us, right? There's been a lot of research on aphid-resistant soybean. Think about the RAG genes, RAG, which there's a lot of research there showing that these aphid-resistant soybean can be effective against the pest, reducing the frequency of outbreaks, magnitude of outbreaks, but the availability of those aphid-resistant soybean lines is pretty limited, and right now pretty much catered just to the organic market. So maybe I'll pause there quick, Anthony, if you've got any other questions or comments about soybean aphid before we switch over.
Yeah, one last, general for soybean aphid could be others, but how about lorsban or chlorpyrifos? What you've seen both for efficacy, but also kind of regulatory updates there.
Yeah, so chlorpyrifos, what a lot of people think of as lorsban, continues to be very effective against soybean aphid, but we've had that regulatory roller coaster ride going on where it was available, it wasn't available. To the extent of my knowledge, there are chlorpyrifos containing products that are available for use in soybean and a limited number of other crops in Minnesota. I think the situation is that these products need to have the new label. And if they have the new label, my understanding is that they can be used. I'm not sure if MDA has officially announced that yet for this year. I think it's kind of a year by year decision. But on their website, they do have a tool for searching which products are able to be used in Minnesota crops.
Yeah, that's what
Have you
I've
heard
heard
any other
too.
updates, Anthony?
Just the short version, I think I've told people too, is if you own something before June 30th last year, that would have been an old label. So existing stock you wouldn't be able to use this year. You need to have a
Right.
new updated label like you're mentioning.
Yeah. Okay, well, with that, let's shift gears and talk about a different pest. So again, this is a newer insect, and as of 2019, it was new to science, right? Nobody knew this thing existed prior to that. It's a tiny little fly, and its larvae live inside the stems of soybean. They're these bright orange colored maggots. The adults are pretty nondescript. If you're walking around in a soybean field that's infested, you're probably not going to notice the adults. But what you are likely to notice would be the larvae. Again, they're that bright orange color, fairly easy to see if you start peeling back some of the stem tissue. And I think I've got some pictures coming up showing the injury that the larvae make, but just a quick overview of the life cycle. The adult flies lay their eggs in the stem, and they're laying their eggs in cracks in the stem at the base of the plant. And then those eggs hatch into larvae. Those larvae progress through 3 different stages inside the plant. Eventually, the larvae will drop out of the plant and then go into the soil, make a cocoon, which you can see in the left of that picture with the soil or sand particles attached to it. Inside there, there's a pupa. And then the adult will emerge to start the life cycle over again. Soybean gall midge is its known range is continuing to expand each year. We're finding it in additional counties every year. In Minnesota, you can see we found it in some additional counties near the Twin Cities, getting all the way out to the Minnesota-Wisconsin border. Some of those, like in Dakota County, were detections in soybean, but then in Ramsey County, and I'm blanking on the name of the county, just north of Washington County, it was in sweet clover, which is one of the alternate hosts, and that's Chisago County up there. So soybean gall midge, as its name implies, feeds on soybean, but it's also been found in sweet clover, and we've been finding it pretty easily in sweet clover around the Twin Cities area. It has been reported feeding on alfalfa and on dry beans, and now some laboratory work that we've got going on with it. We've been finding it can infest and survive in various different legume crops. So getting back to soybean, I mentioned those larvae develop inside the stem at the base of the plant. You get these dark lesions, makes the stems brittle, they'll lodge, plants will wilt and die. Management of this pest, there's not going to be a silver bullet. Like when soybean aphid showed up, we were able to manage it pretty well with insecticides. Most of the work so far has shown that insecticides are not all that effective. Cultural control, our colleague in Nebraska has shown that hilling can be very effective, and this is where you pile up soil at the base of the plant to cover the fissures or cracks where the adults lay their eggs. And you can see in this picture areas that are hilled, where you've got live plants, and areas that were not hilled, where you basically have no plants. So it can be very effective, but it's not a very practical method, right? Most people don't have the implements for, piling up that soil at the base of those plants. Some work we're doing is looking at resistance in soybean to the soybean resistance to soybean gall midge, doing some laboratory experiments, following up on field work that our colleagues have done in Nebraska, Iowa, and South Dakota. And what we've got here is a bunch of different soybean accessions. And you can see that the top two, which are susceptible checks, varieties that we know soybean gall midge will do well on, had more soybean gall midge than some other lines. And this one line at the bottom was actually significantly different from those susceptible. So there's some promise that we could have some genetic material that breeders could use to hopefully get some resistance out there for soybean gall midge. How are we doing for time, Anthony?
We got a couple more minutes presentation time.
Yep.
Maybe at 9:30 we'd want to be wrapped up.
Perfect. So I want to shift into biological control, and that's where my lab's been focusing much of our effort. And we're working on a farm in Lamberton, Minnesota for some of our earlier work. And part of it was focused on predators, where we've been using pitfall traps to figure out which predators occur on the soil. Documented a particular predator called Pterosticus melanarius as the most abundant predator in those fields. And then we followed up with that with some laboratory work to see how many soybean gall midge larvae this predator could consume. And what you can see in this graph is across the bottom is the number of prey that we offered, and across the side of the graph is how many prey were consumed. And this thing was basically eating as many larvae as we were putting into the dish. We maxed out at 60 larvae per dish, and they were averaging about, almost 40 larvae that they were eating in an hour. Some were eating up to 60 larvae in an hour. So they're pretty voracious predators, at least when they're confined to a petri dish. The next thing that we wanted to do is see if they're actually feeding on the pest in the field. So we dissected out the guts of these predators and did some genetic work to see if the predators contained DNA from the soybean gall midge. And if a predator has soybean gall midge DNA inside of its stomach, it indicates that it fed on it in the field. So not only were we able to document that these predators are indeed eating soybean gall midge in the field, we found that the percentage of the beetles that had eaten soybean gall midge increased with increasing soybean gall midge density. So the more soybean gall midge you have out there, the more predators there are that are eating that prey. And then the next group of natural enemies that we wanted to look at are biological control agents was parasitoids. And again, some of you have probably seen presentations where I've talked about this insect, Sinopius maximum, which is a new species that we discovered. You can see how small it is on a penny, about the size of the number 1 on the year of a penny. And then more recently, we discovered an additional species of parasitic wasp attacking soybean gall midge. This one was previously known, but only from Canada, and there was no knowledge of what insects it was attacking or feeding on. So now we've confirmed that this insect, this additional wasp, occurs in the Midwest and that it also feeds on soybean gall midge. So one of our students, Sarah Von Grease, is doing a multi-state project with Minnesota, South Dakota, Nebraska, and Iowa, sampling multiple fields over 3 years and then using molecular tools to determine if the soybean gall midge larvae contain wasp DNA as a way to see if those gall midge larvae have been parasitized or attacked by these different parasitoid wasps. And the molecular test is refined enough where we can distinguish which species of wasp has attacked the gall midge larvae. And here's just a quick summary table for those 4 different states, the 2 different species of wasps, Sinopius maximum, Sinopius ruficoxum, and then just a snapshot of 2 of the years of data, 2023, 2024. We can see that in all states, we've detected some parasitism by these wasps. And in Nebraska, we've found, pretty high levels of parasitism. In some fields, up to 50 to 60% of the soybean gall midge larvae have been attacked by Sinopius ruficoxum, the wasp with the very long, skinny abdomen. So that's very promising for biological control. A lot of times we don't see levels that high. So just try to wrap things up here. Soybean gall midge, it's a new pest, something to be on the lookout for. Across Minnesota, most of our fields have fairly low infestations when we find it. Bruce Potter and his team have had to work pretty hard to find this insect in fields. The plants aren't always very symptomatic. If you're going to be looking for it, look for the wilting of plants, death of plants on the field edges, and then look for those kind of necrotic lesions at the base of the plant. Peel open those tissues, look for the orange larvae. And again, there's going to be no easy button for managing this pest. We're going to need true integrated pest management where we're incorporating multiple different management tactics. So I'll just kind of conclude here by pointing out that we have an extension webinar coming up soon, a multi-state webinar. That'll be March 19th in the morning. And there's a QR code here, the Z link, where you can get to the registration. And here's just a screenshot from last year's. We're going to have presenters, a lot of graduate students from the multiple states presenting updates on the latest research on the biology and management of this insect. So I think with that, Anthony, I'll turn it over to you to see if there's any comments or questions.
Yeah, a couple questions came in. I might save some till towards the end of the session here, but one of the ones I was thinking about is, let's say you're going to have some growers who might be concerned, especially because we don't have rescue treatments, so they're worried about risk looking ahead. And whether it's for a grower that maybe hasn't had issues yet, or folks maybe in Southwest Minnesota where it's definitely around, they might be worried about it. You have those 2 groups. So how do you kind of address this kind of risk issue to folks and kind of what they maybe could be doing now or keeping an eye out for?
For soybean gall midge?
Yep, soybean gall midge.
I think very, very few growers should be concerned about spraying for this pest in Minnesota. We just haven't seen that many fields with heavy infestations. We've had to work pretty hard to find infestations to do our research. So in presenting this, I don't want to be scaring people into thinking they need to be spraying their fields. As infestations increase, or in those very limited areas where there are heavier infestations, and some of the stuff that's going on in Nebraska, some people are spraying. I think the challenge is going to be timing of those sprays, because we've got multiple generations of this insect. Those generations, the adults emerge over a fairly long period of time, so it's probably going to require multiple applications to get protection. Yeah, I think it's going to be tough with insecticides unless we can figure out an approach to timing those applications better.
I'll take one more question, soybean gall midge related, then we'll move over to the corn side of things. How about, you mentioned other hosts. where did soybean gall midge originally come from? I can't remember if you covered that one or not, but you want to just mention kind of, is it
Yeah.
native, not native?
Yeah, that's a good question, right? It's a new species. We honestly don't know where it originally came from. In a sense that we don't know if it's originally from North America or if it came somewhere else as an invasive, came from somewhere else as an invasive species. There's no records of it occurring anywhere else outside the Midwest. The fact that we're finding soybean gall midge feeding on more and more plant species kind of leaves me thinking that maybe it is a native species. Maybe it was just feeding on some native legumes that nobody paid much attention to, let alone, little maggots that were feeding on those plants. And for whatever reason, it underwent some kind of a host range expansion where it can now feed on soybean, but we don't know for sure yet.
All right. Thanks, Bob.
Yep.
We'll move on to Faye, if you want to bring up your slides and we'll talk about what's going on the corn insect world.
Okay, good morning. So let's move to some corn insect pests. So today I would like to give some update about the Bt resistance in European corn borer populations during last years we sampled in Minnesota and Wisconsin. So a little bit background about this old insect pest, and we have new problems right now. So it's a Lepidoptera insect pest, and currently European corn borer is a major target of transgenic Bt corn. And in Minnesota, we have two types of this pest. One is univolting, and by its name, it means we only have one generation per year. So this type is predominant in the north area. And we have another type, multivolting, and that means we have two or even more generations per year. So depending on the temperature. So in some area of Minnesota, we have these mixed types, univolting and multivolting. So as a Lepidoptera insect pest, it has typically 4 different stages for its life cycle. So the adult lays eggs on the underneath of the corn leaf. And once these eggs hatched, they will initially feed on the leaf tissues and then move inside the stalk. And they complete their life cycle for the larva stage inside the stalk and become pupae. Sometimes some of them can be inside, and most of them will get out of the corn stalk and become pupae in the soil. So here are some typical damage for the corn borer. So they will do the the stalk breakage. So don't confuse this kind of damage with the rootworm damage. So rootworm damage, you have some the larger plants, especially near the base of the plants. But for corn borer damage, you will see the plant is breaked in the middle of the plants. And this is a picture I took from the field. And the growers thought it was initially was the rootworm damage, but when we traveled there and we found it's not, it's like a European corn borer damage. So here are more pictures about the damage of the European corn borer. Because why do we need to mention this? Because this is old pest, and normally we don't see this kind of damage in the field currently. So if you look at the field and if you find some leaf tissue damage, like this array of holes that are probably coming from corn borers, and once they get bigger, they will fit in the wall and move inside the stalk. And you will see some entrance or exist holes in the stalk later in the season. So another thing I want to mention is another species, like stalk borer. So this is a common pest and also damaging corn in the fields. And also we see this in the fields in Minnesota and the other states in the corn belt. And also we have one generation per year, but it has not like European corn borer. We have typically 5 different instars for the larva, but this one we have 7 to 16 different instars. How it is very difficult to distinguish these 2 different insect pests, especially for the larva stage. You can see these insects, they are black or brown, and especially when they are young, you can see this purple brown band along its body. And these insects initially feed on the grass, and once they get bigger, they will move into the corn plants during June and July. So if you go to your field, you see this kind of damage, probably initial thought that's European corn borer damage. But this is what happened this year. We went there and we saw lots of damage. We tried to collect some populations, but when we split the corn and we found it's not European corn borer and it's less common stalk borer. So especially as I mentioned, these insect pests initially they feed on grass, and then when they get bigger, they move to the cornfields. So it is most common in the first few rows. So if you go to the middle of the field, probably you don't see this kind of pest. And another thing you need to pay attention. So transgenic Bt corn, as we showing here, CRA1, CRA2 Bt proteins are not effective. And these are the plants we sampled from the field. And all these are the Bt corn plants, and we see lots of damage. So if you plant Bt corn and you see lots of this kind of damage, so probably it's like a common stock borer, because like a European corn borer, we don't have so much like damage in Bt corn fields right now. Okay, so let's move back to our today's pest, European corn borer. So this pest actually is the first target pest for like a Bt corn since 1996. And the first we have BT11, like Yearguard, CRA1AB, and then 2023 we have Hercules with like a CRA1F Bt protein, and in 2020 we have CRA1F 1105 and CRA2AB2 together expressed in Bt double pose smart stack these corn hybrids. And so far this transgenic Bt corn has provided excellent control efficacy for almost 3 decades for management of this pest. So if you look at the population density, like before and after the commercialization of this transgenic Bt corn, so red circle one are the population density after 1996, and we use the transgenic Bt corn, we can see the population density is significantly lower compared to those like not so-called with red. Those are populations before 1996 in these like 3 different states. So why do we need to care about this population right now? Because of resistance. The big threat for the long-term use of these kind of transgenic Bt technologies is the evolution of resistance. And in 2018, in our neighboring country, Canada, the scientists found the CRA1F resistance for the first time in Nova Scotia. And during 2019 to 2020, and they found the CRA1F resistance in this pest has expanded into other areas in Canada. And in 2023, they found that this resistance not only to CRA1F, but also to other CRA1A15, CRA1AB, and CRA2AB2 Bt proteins in Canada. And even worse, during 2023 and 2024, the scientists in Connecticut, and they found a practical resistance of European corn borer to CRA1AB, CRA1A5, and CRA2AB2, all these technologies in sweet corn fields. And this marked the first case of Bt practical resistance in the mainland US. And they just published a paper in Journal of Economic Entomology last month. So if you are interested, you can go back and read this paper. So if you look at this left picture here, and the red circle one are the already documented resistance for like European corn borer in Canada, and the blue circle one is the population for the resistance documented in Connecticut. And if you look at these like spots for resistance, they are close to like US, especially for the Manitoba one, close to the Minnesota and the Corn Belt. And plus the mouse of European corn borer, they can travel long distance, any distance from like 12 kilograms to more than like 720 kilograms. So it is a high chance these like mouse resistant insects can travel long distance to the corn belt. And also think about the high adoption, about 90%, like in the field, we planted like a Bt corn, and the long-term use for almost 3 decades. So that means we have strong selection pressure for the Bt resistance in European corn borer population. And we put all these together, so it is a high risk of this pest to evolve resistance like in the corn belt. If we look at our like a field like sampling data, so this is the data from 2024 with our like collaborator Crystal from Wisconsin, and they sampled 162 fields in 2024, and they found one field with like a damage about 20% with one plant with live insects, but this plant is Bt plant. It expresses the CRA1AB. That means we already have resistance in the field, but it may be low right now. So this is the ECB like field survey in Minnesota in last year. So in this field, we found some Bt plants damaged by like corn borers, and we tested using strips and confirmed the Bt expression. And in nearby plants, and we found some European cumber. If you can see here, like on the left, the bottom like a picture, it has like a European cumber larvae inside the cone ears. So we know these are European cumber here, and they also cause damage. That means we all have some resistance genes in the field. And if we look at some fields, especially for those like non-BT continuous cone fields, we still have large populations of European cumber. So these are the samples we did the last 2 years from North Dakota, Minnesota, and Wisconsin. And if you look at the field, we found like 8 to 90% of plants with like European cumber damage, and we can easily sample more than 1,500 insects from these fields. So that means we still have large populations and some hotspots in the field. So this is my student's like Tatum. So she did like lots of like study. So what we do, we collect the insects from the field during 2023 to 2025. And after that, we rear these like larvae in the field and break the diapause. And we do the single pairing until the second, we call it like F2 generation. And these insects, F2, we will test them against all the CRA1-BT proteins, CRA1F, CRA1AB, CRA1F5, and CRA2AB2 at a diagnosed concentration. So if we have any survivors on these like BT proteins, that means we have resistance genes in the field. Otherwise, they are clear. So if we look at the results here, first we look at the first mode of action, CRA1. We have CRA1F, CRA1AB, CRA1F5, and we test a total of 84 populations, and we didn't find any survivors to third instar. That means we don't have any major resistance alleles out of these 84 populations. So that means the CRA1 resistance right now in the field, sampled from Wisconsin and Minnesota, is low. However, if you look at the other mode of action, CRA2AB2, and out of these 84 populations, we found 14 populations shows like higher like survival. That means the CRA2AB2 resistance probably is high in the field right now. So we established these like the survivors from the like the bioassay, and we try to document what's the resistance level for these populations. So if you look at this figure, the top one is a susceptible population. At like concentration 360, we found the mortality is 100%. However, for these field populations sampled from Wisconsin and Minnesota, even at the highest concentration, 5,400, we only have 0 to like 27, 28% mortality. That means all these field populations, they show high level resistance to CRA2AB2. So we do the full range bioassay, and also we calculate the LC50. That means the concentration can kill 50% of the insects in the population. And we calculate resistance ratio compared to our first like susceptible population. The resistance ratio is more than 38.7 fold. So any resistance ratio more than 10 for BT resistance, that means the population is resistant. So these resistance ratio clearly document the populations sampled from the field for this CRA2AB2, they are highly resistant. So here we summarize the population, why we found the resistance for like Wisconsin, like Minnesota. It's widespread. That means the CRA2AB2 resistance right now is widespread in these two states. So from that study, we know the CRA1, it seems still very effective, and the CRA2AB2 resistance widespread. So right now we have the BT hybrids express both CRA1 and the CRA2-BT proteins. How about these CRA1 proteins? Whether they can control the CRA2AB2 resistance. So we did some cross-resistance study, and we used our resistant insects and tested them against all 3 CRA1 proteins. And we found the resistance ratio is low. As you mentioned before, if a resistance ratio is less than 10, that means we have no cross resistance. And all these CRA1-BT proteins are very effective for managing the European cumber populations. So that's the assays in the lab. And we wanted to know whether these insects could survive on the whole plant, especially for those commercialized BT cone hybrids. So we did some whole plant tests in the greenhouse as well as some leaf tissue tests. So in this test, we use the non-BT and some like experimental hybrids. These like compounds only express one CRY1AB, CRY15, or CRY2AB2 BT proteins. And also we included one commercialized BT con hybrid, the BT double pole, which expresses both CRY1 and CRY2 BT proteins. And we used susceptible, resistant, and the crosses between the susceptible and the resistant insects. We call the heterozygotes as eyes. And we infest 8 insects into each plant, and we record data after 28 days. So if you look at the data here, the y-axis are the percent plant with live insects, and the x-axis are different cone hybrids. So if we look at the non-BT, the red one is the susceptible insects, black one is susceptible insects, the red one is the resistant insects, and the blue one is heterozygotes. The cross is resulting from the susceptible with like a resistant. And on non-BT, we can see 8 to 95% of the plants with live insects. That means all these insects could survive very well on the non-BT. However, if we look at the 2-CRY2, AB2, one is low concentration and one is high concentration, we found the resistant insects, the red one, survived very well with no differences compared to the non-BT. However, we also have some heterozygotes could survive on this CRY2AB2. That means the resistance may be not like recessive. It could be like partially dominant. However, if we look at the survival or like the number of insects on the CRY1 proteins, CRY105 and CRY1AB, we didn't find any insects. And finally, if we look at the commercialized VT double pole, and we didn't find any insects, that means any CRY1 proteins right now in the plants can still kill those CRY2AB2 insects. And these are some like leaf tissue assays. So the y-axis are the mortality, and the x-axis are the non-BT and the BT double pole that commercialized BT con hybrids. And on non-BT, we can see these insects on the left picture. Right picture, they survived very well. However, on the BT double pole, even for these like orange one, these are resistant insects, we have 100% mortality. So the summary for the resistance for this insect pest, based on our study over 2 years, we found resistance to all the CRY1, CRY1F, CRY1AB, and CRY105 is low. That means all these proteins can still be very effective for managing the European cambour populations in the field. However, the resistance to CRY2 is very high. And the good news, these CRY2AB2 resistance populations can still be killed by the CRY1 BT proteins in the con plants. However, if we look at these commercialized BT con hybrids, BT double pole, Triceptor, Polka, Vorsid, or Smart Stack, all these con hybrids, they have both CRY1 and CRY2, and right now, the CRY2 is not effective. That means we only have one mode of action in these con hybrids. So that means selection pressure would be high for this mode of action. And if we think about the situation in Canada, so it is just opposite. So resistance widespread to all the CRY1 proteins, but CRY2AB2 is very effective. As I mentioned, these populations in Canada, they can migrate long distance if these populations for CRY1 resistance migrate into Minnesota, Wisconsin, or other corn belt states, and they mate with our CRY2AB2 resistant population. That can generate resistance insects resistant to both CRY1 and CRY2. And in that case, we will have trouble for the resistance management. So with that, I would like to thank my group, my postdoc, my undergrad students, and my grad students, as well as like the collaborators for ECB collection and the funding supporters from these projects. I think I have 10 minutes for questions.
Yeah, thanks, Fei. So I actually had a couple thoughts kind of on one of your previous slides too, talking about migration. So when we have a resistant population in Canada to one trait and the other trait here in Minnesota, we're finding it. Do you know or have you heard how quickly populations can migrate? If we have that population in Canada, will it be a few years potentially till potentially those populations would find their way down here, or how quickly does that potentially happen?
So the insects So right now, based on the it's not like a document, but based on the hypothesis, the Connecticut population resistance that we observed during 2023 and 2024, it's a high chance these populations can travel from the initial resistant population in Canada from Nova Scotia, because if you look at this big circle here, it is very close to Connecticut. And if you look at these migrants as shown on this right picture, it can travel long distance. So it depends on the weather. If you have a storm on this direction, so the insects can travel a few hours. In 1 day, they can be here.
All right. Kind of another follow-up is, a lot of folks, especially my age, I never had to deal with European corn borer as a kid on our farm. But if you're kind of another generation older than me, you probably remember it pretty well. But do you want to remind folks, if these traits really aren't effective anymore, what are our management options after that? What do you have to do to try to manage this, and how challenging is that?
So right now, I don't want to scare, because right now all the corn hybrids in the market, they contain both CRA1 and CRA2, and we have some problem with CRA2, but CRA1, as I mentioned in my study, they are very effective. So we don't need to worry about the efficacy right now in the field. However, from the standpoint of resistance, once the insect develop resistance, they can do very fast. And once they establish resistance population, and the efficacy probably significantly reduced. However, based on the current information, we don't have any good traits in the market available in the very 10 years. If we look at the old the management strategies, we can use a catch practices, and also we can use the insecticides. I think the most one is the insecticides, but the scouting is the most important. As I mentioned, the insects, the larvae, they will feed on the leaves only for around 10 days. So you need to do a very good timing for scouting. Once they finish the leaf feeding, they will get inside the stalk. So if you miss this kind of management window time, they move inside, the foliar insects cannot reach the insects, and you don't have any efficacy. So if you try to use traditional way, do scouting and use those pyrethroid insecticides, should be good enough.
All right, I have a couple of questions. I'll try to rope it into one here. This could also apply to corn rootworm species as well as European corn borer, but about the folks thinking about planting non-BT corn, is that a viable strategy? When might there be risks for that? How should they weigh that decision?
Okay, so if you use a non-BT corn, it depends. So the first thing, you need to understand their fields. What is the major pest over there? So if you have corn borer populations, as I mentioned, so you still have a hotspot over there. So it's especially for continuous corn, and you need to do the scouting. And if you have some problems and you don't have BT, you need to use foliar insecticides, as I mentioned before. If you plant non-BT corn for management of corn rootworm, so same thing, you need to do scouting, and the best management for corn rootworm is crop rotation. So if you have the insects over there, and this year you plant corn, and the next year you rotate it to soybean or another insect pest, probably you can rule out all the corn rootworm eggs, and that can help you management. But if you have extended diapause of northern corn rootworm, that may be another situation. So you need to think about what kind of species in your field.
All right, thanks, Fei. I think for both of you, Bob and Fei, one of the questions that's come up a little bit are cutworms or true armyworm. I know, Bob, if you want to mention a little bit, we were talking a little bit earlier just about how soybeans actually can get hit by true armyworm sometimes, but maybe not as common, but especially in the cover crop situations. So you kind of want to mention what's going on with cover crops and insects a little bit?
Yeah, there's a lot of interest in trying to increase cover crop adoption, and I think any big changes we're making to our production systems, that could potentially affect the insect pests, right? So we've done some research where we've shown that planting soybean, like relay cropping, into a fall planted rye cover crop can actually result in lower infestations of soybean aphid. So somehow having that other plant species there affects the ability of the aphids to either colonize or stay on the soybean. More recently now, we're doing work with some of the newer cover crops that are being developed, like pennycress and camelina, and we're seeing a similar pattern where relay cropping soybean into those can result in lower pest pressure. I think one of the challenges is going to, fall to the agronomists of how to tweak that production system to reduce competition between soybean and whatever the cover crop is, right? So we don't have, a yield drag coming from that. And then, we could get these additional benefits of potential pest suppression. I guess what you're referring to, Anthony, is kind of the flip side of that, right? Sometimes having that cover crop there can create pest problems. It's not guaranteed that you're going to have a pest problem or get pest suppression from it. But pests like true armyworm can be attracted to that, those dense grass cover crops. And I think it's a bigger concern for corn. But, Anthony, you've shared pictures of soybean field that was ravaged in the early vegetative growth stages when it got hit with a big influx of armyworm. Fei, maybe you can share a little more on the corn end of it.
Yeah, for cover crop in corn, I think the most important is to do the terminate timing. So normally we do 2 to 3 weeks termination before we plant corn. So in that case, the hatched larvae they don't have food and they will be dead. And even if you have a few population, so the corn, if you have low population, the corn after the insects can feed in, the corn can recover very fast, not like soybean.
Yeah, I think that's a good point, right? When that cover crop is terminated, so that the potential pests there can starve before the corn or soybean starts growing. In looking for that potential for pest suppression, like we're seeing with soybean aphid, I think that terminating earlier would probably decrease the likelihood of seeing those benefits of pest suppression, unfortunately. Yeah.
Great. I think I have time for just one question. I think we had quite a few soybean gallmage questions come in later, Bob. So I'm going to rope a couple in for you here.
Okay.
One, can it survive the winter here? Or how cold does it take? The other part was timing of it a little bit more, just reiterating when does it show up and also when is healing effective? A couple questions on that.
So the timing of infestation, again, the adult soybean gallmage, the flies rely on cracks in the stem for laying their eggs so the larvae can get inside the stem. And at the base of the stem, those cracks start naturally appearing around the V2, V3 growth stage. Some of the work out in Nebraska then is showing that hilling needs to be applied pretty early, probably like V2 to V5. The earlier the better to protect those plants, before the eggs and larvae can get in the stems. As for the effects of cold temperatures, we are doing some research there. We just had a scientific paper accepted for publication looking at short-term exposures to cold. And it looks like soybean gallmage can handle pretty well. Pretty cold temperatures where we were, it had to get down to -13 Fahrenheit before we started seeing big increases in how many of the gallmage were actually freezing and dying. But some more recent work, we're looking at longer durations of exposure to the cold, which would probably be a more realistic scenario. And we're seeing that they're much more susceptible to cold when that duration is increased. So even at 26 Fahrenheit, right? So just a little bit below freezing, we're seeing pretty high mortality happening after, a few days, and almost complete mortality at 5 or 6 days.
All right. Thanks, Bob. I think it's time for us to wrap up here. So thank you again, everyone, for attending today's Strategic Farming Let's Talk Crops program. These sessions are brought to you by University of Minnesota Extension and with generous support from the Minnesota Soybean Research and Promotion Council, as well as the Minnesota Corn Research and Promotion Council.